So today our topic is nervous tissue. And now let's revise diagram which shows types of tissues, classification of tissues. Here we can see that tissues are divided into borderline tissues, epithelial tissue, and inner tissues, including connective tissues or tissues of inner environment, blood and lymph, connective tissue proper, and skeletal tissues, and specialized tissues which have special properties of their functions and structure. They include muscle and nervous tissue. And today we will talk about nervous tissue, about main components and structure of nervous tissue. So here we can see a histological slide which shows us structure of nervous tissue. It's a basis of nervous system. So all organs of nervous system, they are composed of nervous tissue. And here we can see main structures of nervous tissue or main cells of nervous tissue. They are nervous cells or nerve cells, or also they are called neurons. So neuron or nerve cell or neurocytes, they are main cells in the nervous tissue. It's easy to remember. And neurons or nerve cells or neurocytes, they provide regulation of functions of another cells and organs. They generate and send nerve impulses, and target cells, they receive those nerve impulses, and there is special conduction of information. There is mechanism which includes neurotransmitters, special chemical substances, and later we'll discuss how it's realized. So neurons, they generate and send nerve impulses, and they directly run to target cells which accept this nerve information. And here we can see neuron, main cells of the nervous tissue, and we'll discuss their structure. And also nervous tissue includes another family of cells, another set of cells. They are called glial cells or glia. There are cells, here we can see, which surround nervous cell. Nervous cell is shown as green, and red points, red star-like structures, they are glial cells. What is glia? Glia is supportive part of nervous system and nervous tissue. Neurons or nerve cells, neurocytes, they provide main functions of the nervous system of nervous tissue. They generate impulses, they provide regulation of some cells and organs. And glial cells, they provide support and they maintain neurons. So neurons, they are main, but they need support. And this support is provided by glial cells. They provide nutrition, they provide protection, they maintain neurons, they provide defense, and all other non-specific functions are provided by glial cells. So we have this division, two groups of cells. First group, it includes neurons. They are specific cells which provide functions, main functions of nerve system and nervous tissue. And glial cells, they help neurons and they provide support. So there are supportive cells. And here we can see neurons, big cells, and they are highly specialized. And also there are glial cells which surround neurons and they provide support for them. And also here we can see neurons, and there are numerous glial cells. There are different kinds of glial cells, and today we will talk about them. What are the differences between them? So let's begin from neurons. Here we can see nervous cells or neurons. They are the main cells in the nervous tissue. And here we can see also nuclei of glial cells which surround neurons. So let's begin from neuron or nervous cell. Here we can see structure of nervous cell or neuron, and it has cell body, or it's called perikaryon. Karyot means nucleus, so perikaryon is those part of the neuron which contains nucleus. It's nucleus and cytoplasm which surrounds this. So neuron, generally it has star-like or branched shape, or they may have different shape, but we can see that they have cell bodies or perikaryons, and also they have processes or projections. Here we can see long processes which begin from perikaryon, from cell body of neuron. So here we can see two principal parts of neuron. So cell body or perikaryon, it contains nucleus, it contains all necessary organelles, all general organelles, and also there are special organelles. We will discuss them. And what are the processes? Processes, they begin from perikaryon, and also they contain some amount of cytoplasm, and they are surrounded with plasmalemma, with cell membrane. So cell membrane covers processes, it covers perikaryon, and here we can see this branched shape of the cell, of nervous cell. And processes, they have nerve endings. Here we can see nerve endings, and they provide communication between the neighboring cells, and also they may receive information from outer environment or from inner environment, and also they may send information to another cells, and we'll discuss this. So perikaryon and processes, or cell body and processes, they are principal parts of the nervous cells. And what are the processes? We have two principal kinds of processes. There are dendrites and axons. Usually, cells have only one axon, and cells, they usually have different number of dendrites. So if cell has processes, one of them always is axon, and all others are dendrites. Axon, it's only one, and it always is present. Dendrites may be absent in some cases. Dendrites, number of dendrites may vary from 0 to numerous. So cell may have 1 dendrite, 2 dendrites, 3 dendrites, and more. So here we can see this cell, it has 4 dendrites and 1 axon. So 1 axon and 4 dendrites in this cell are present, but number of dendrites may be different. Dendrites may be absent, 1 dendrite may be present, 2, 3, and more dendrites may be present in the cell. So it's numbers of the dendrites and axon. What is the difference between them? The main difference is functional difference, because all processes, they provide, they carry impulses in some direction. They are needed to provide communication between the cells. They have long processes and they communicate with each other. They form network, and processes, they bring nerve impulses. So what is the difference of direction? Dendrites, they carry impulses to the perikaryon. They are processes which bring impulses to the cell body. And axon, it receives information from cell body and carries it to the peripheral parts, to another cell or to the target cells. So it brings away from the cell body, it brings nerve impulses. It's main difference between dendrites and axon. So dendrites, they carry impulses to the cell. Cell collects impulses, collects information from different sources. It provides processing of the information, and all information goes to one axon, which brings this impulse to another cell or to target cells. It's main principle of the functioning of neurons. But also neurons, they may have different structural differences. They may have different number of dendrites, of axon, and it reflects their functional specialization. Dendrites may be long or may be short. They may be branched or unbranched, as well as axon. Axon usually is long, but in some cells axons may be short. So it's not the principal difference between dendrites and axon. The main difference in dendrites and axon is direction of nerve impulse. And here we can see different cells, different neurons, and its morphological classification of neurons. And here we can see that neurons, they have different number of processes. So here we can see perikaryons of each cell. Here we can see perikaryons, cell bodies which contain nucleus, and we can see here that each cell, it has one axon. This cell has one axon, this cell also its axon, this cell has axon, and this cell also has axon. But let's look at number of dendrites. This cell hasn't dendrites. This cell, it has one dendrite. This cell also has one dendrite, but dendrite and axon, they have common trunk, common beginning. And this cell, it has 5 dendrites. So morphological classification is based on number of dendrites and axons. We classify neurons into 4 groups. We call them unipolar, bipolar, pseudo-unipolar, and multipolar neurons. Unipolar neurons, unit means one. They have only one axon, only one processes, only one axon. Dendrites are absent. That's why they are unipolar, one pole, one processes. Bipolar neurons, they have 2 poles. One is dendrite, another is axon. Pseudo-unipolar neurons, they are bipolar, and sometimes they are described as kind of bipolar neurons. But what is the difference between bipolar and pseudo-unipolar neurons? In pseudo-unipolar neurons, dendrite and axon, they have common beginning, common trunk. It begins from perikaryon. And bipolar neurons, they have distinct beginnings of dendrite and axon. But why do we call this type of neurons pseudo-unipolar? Because they are alike unipolar neurons. They have one processus which begins from cell body, but here this processus is divided into dendrite and axon. That's why we call them pseudo-unipolar. So they are alike unipolar, they are alike bipolar, but they exactly are pseudo-unipolar, third type of neurons. And some neurons, they have 2 and more dendrites, and they are called multipolar neurons. Bipolar neurons, they have 2 processes, 1 dendrite and 1 axon. Multipolar, they have 1 axon and 2 and more dendrites. They are called multipolar. They have numerous processes. So we have unipolar, bipolar, pseudo-unipolar neurons, and multipolar neurons. It's morphological classification. And where they are present, in which structures. Unipolar neurons usually are present only during embryogenesis of nervous system, and they aren't found in adulthood in our organs, but sometimes rods and cones of retina of eye, they are described as unipolar neurons, sometimes as bipolar neurons. Usually, clearly, unipolar neurons are present in the embryogenesis, during embryogenesis in embryos. Bipolar neurons are present in human retina of eye, and also they are present in some ganglia. For example, in hearing ganglion, there are bipolar neurons. So they usually are present in sensory organs. Pseudo-unipolar neurons, they are present in spinal ganglia. There are spinal ganglia near spinal cord, and they contain pseudo-unipolar neurons. They are present there. And the most numerous neurons are multipolar neurons. And central nervous system, it contains mainly and almost all neurons in CNS are multipolar. They are the most numerous neurons in our body. So CNS, it's composed of multipolar neurons. Pseudo-unipolar neurons are present in spinal ganglia. Bipolar neurons are present in some structures of sensory organs, and unipolar neurons are present in during embryogenesis. And multipolar neurons also are present in the ganglia of autonomic nervous system, and we'll study them. We'll discuss them in topic nervous system. So it's morphological classification for principal types of neurons. And also we have functional classification of neurons. It was morphological classification which takes into account appearance of neurons, and according to the functions we divide neurons into 3 principal groups. There are sensory neurons, there are association neurons or interneurons and motor neurons. So what is the principal difference? Sensory neurons, they take information from outer environment and bring this information to CNS. For example, here we can see a cross-section of the spinal cord, and here we can see a spinal ganglion. It's present near the spinal cord, and there are sensory neurons. They have long dendrites and they are present in the peripheral nerves, and they uptake information from outer environment. For example, if you touch something, for example, a cup with hot tea, you feel that it's hot, and your dendrites send information to spinal ganglia, to cell body of sensory neuron. It provides processing of the information, and axon of the cell brings information to CNS. So it's sensory neuron or efferent neuron. And next neuron, it's interneuron. It receives information from previous neuron, for example, from sensory neuron, and sends information to another neuron. Number of interneurons is different, and in CNS, the most numerous neurons are interneurons. They provide processing of information. They form networks inside CNS and in other organs of nervous system, and finally they send final information to motor neurons. So interneurons, they receive information from another neuron and they send information to another neurons. They are located between neurons. They receive information from neurons and send information to neurons. But sensory neurons, they send information to neurons, but they receive information from outer environment. It's principal difference. And motor neurons or efferent neurons, they send information. They usually have long axons, and they are going to the peripheral parts of nervous system, and they are sending information to effectors, for example, to muscles or glands. So when you touch hot cup of tea, your dendrites of sensory neurons, they bring efferent information to cell bodies of sensory neurons. Sensory neurons, they send information to interneurons. Interneurons, they send information to motor neurons, and motor neuron send information to your muscles, and you don't touch this hot cup of tea because it's dangerous. It's called reflex arc, and here we can see three principal kinds of neurons. Sensory neurons, they communicate with outer environment via dendrites. They uptake information. Interneurons, they communicate with another neurons, but they haven't contact with outer environment. They form networks inside a nervous system. And motor neurons, they receive information from interneuron or from sensory neuron and send this information to effector organs, which are under regulation of motor neurons, to muscle cells, to another glandular cells. So they react somehow to action of motor neurons. It's functional classification of neurons. So we have sensory neurons, interneurons, and motor neurons. Or sensory neurons are also called efferent neurons, and they provide efferent transmission. It goes to the CNS. And efferent transmission and efferent neurons, motor neurons, they bring impulses from CNS. It's efferent neurons and efferent transmission. And also processes and nerve fibers may be efferent, which carry impulses to cell bodies to CNS, and efferent, which carry impulses from CNS, from cell bodies to the peripheral organs. It's functional classification of neurons. So here we can see a picture which shows us inner structure of neurons. They have special organelles, they have general organelles. So general organelles are present in all cells, and also neurons, they have special organelles. Those special organelles are called neurofibrils. Here we can see neurofibrils, they are some kind of cytoskeleton of nerve cells. And they are specialized, and they are visible here as dark lines in cytoplasm, and they form three-dimensional network inside the cells. So they provide maintenance of the shape of neurons, and also they provide axon transport of substances inside the cells and inside the processes, and they provide essential functioning of the neurons. So they provide formation of three-dimensional network inside the nerve cells, and they help them to provide their functions. Here we can see neurons and those dark three-dimensional network. It's cytoskeleton of neurons, which is called neurofibrils. Here we can see nuclei and cytoplasm. It contains numerous neurofibrils. And in other cell organelles, special organelles which are characteristic for neurons, are chromatophilic substance or Nissl bodies. Here we can see neurons with special Nissl staining method, which includes staining with methylene blue. And here we can see blue granules in the cytoplasm of neurons. Here we can see cell bodies of neurons, their processes, and here we can see blue granules in their cytoplasm. So what are those blue granules? They are places where rough endoplasmic reticulum is present, and usually cisterna and membranes of rough endoplasmic reticulum are located in distinct places. They are tightly packed, and they have appearance of granules with special staining of Nissl method. So Nissl staining, it reveals places of protein synthesis inside neurons. Neurons, they need a lot of proteins to provide their functions. They also should produce neurotransmitters, structural proteins, and here we can see numerous granules. They correspond to places of localization of rough endoplasmic reticulum. And here we can see those places. It's usually called chromatophilic substance or Nissl bodies because Nissl staining method was applied. So what do we see here? We can see cell bodies, dendrites and axon of each cell, and here we can see that axon may be distinguished from dendrites because it hasn't Nissl bodies. Nissl bodies are absent in axons. They are present only in dendrites and cell bodies. So it's a special feature of axons of neurons, and if we apply this staining method, we can distinguish axons and dendrites in nerve cells. And place where axon begins, part of the perikaryon, beginning of axon hillock. It also hasn't Nissl bodies, axon and beginning of the axon, and we can distinguish them. Special organelles of neurons are neurofibrils and Nissl bodies or chromatophilic substance. They are special organelles of nerve cells. And here you can see general organization of a nervous system, and nervous system, its brain, its spinal cord. They have gray matter and white matter. Here you can see in brain, gray matter covers brain, and also some inclusions are present inside the brain as ganglia and nuclei. And in spinal cord, gray matter is inside and it's covered with white matter. White matter here is outside, and in brain it's inside. So what is the principal difference between gray and white matter? It's that gray matter, it's composed of perikaryons of neurons and some and also it includes glia and beginnings of the dendrites and axons. White matter, it's completely composed of axons and dendrites, and perikaryones are absent here, but also white matter, it contains glial cells. So white matter, it hasn't perikaryones, and its principal difference. Perikaryones of nerve cells, of neurons, they are present only in gray matter of brain and spinal cord. And also there are numerous glial cells, and they are present in both in gray matter and white matter. And here we can see different kinds of glial cells. So what are the kinds of glial cells? We divide glia into two principal groups, into microglia and macroglia. Two big groups. Macroglia, it includes some another kinds. It includes astrocytes, oligodendrocytes, and ependyma. And also there is microglia. So what are the kinds of glial cells? Let's begin from astrocytes. Astrocyte is kind of macroglia, big group. Astrocyte, here we can see it provides support of neurons, it maintains them, and also it provides nutrition of neurons. Here we can see blood vessel or capillary, and astrocytes, they have food processes, they surround blood vessels, and on one side they maintain blood vessels. And they uptake some nutrients from blood vessels and bring them to neurons. And on another side, they provide maintenance of neurons. They maintain them, and also they have food processes which surround neurons. So its main function of astrocytes, they provide structural maintenance, they provide structural function, and also they provide nutrition of neurons, and also they provide formation of blood-brain barrier. Blood vessels, they may contain different dangerous substances which may be harmful for neurons. There are different toxins, different viruses, bacteria, etc. And astrocytes, they completely surround blood vessels. They have food processes which surround blood vessels, and they filtrate. They provide selection of substances which may pass through this barrier. So blood cannot freely enter in the tissue of brain, of spinal cord, because there are astrocytes, and they filtrate substances which are going to neurons. Its main functions of astrocytes. And here we can see neurons and astrocytes. On one side, they have feeds or feed processes. They surround capillaries, and they provide this barrier, blood-brain barrier. And on another side, they maintain neurons, and they send to neurons nutrients. They have uptaken them from blood, and they send them to neurons. And there are two kinds of astrocytes, protoplasmic and fibrous astrocytes. Protoplasmic mainly are present in gray matter. Fibrous, they have longer processes, and they are present mainly in white matter. So astrocytes, it means astra, it means star, so they are star-like cells, but they aren't neurons. They are like neurons, but they aren't neurons. They provide support for neurons. They are very important kind of glial cells. And here we can see capillary and astrocyte and food processes and feeds, and they surround this capillary, they filtrate substances from the capillary, and they provide formation of blood-brain barrier. And here we can see capillary, which is surrounded with and feeds of astrocytes, and its blood-brain barrier, which includes endothelial cells of capillaries, basement membrane, and also and feeds of astrocytes. Another type of Glial cells are there are oligodendrocytes. Here we can see oligodendrocyte, and there are neurons, cell bodies of neurons. They have long axons and they have special covering. It's called myelin. Myelin is covering of axons. And we'll talk about this later, what is myelin. But here you can see oligodendrocyte, special glial cell. It has long processes which are rotated above axons of nerve cells. So this oligodendrocyte, it provides formation of myelin in CNS. Oligodendrocytes, they isolate neighboring axons from each other, and also they are necessary for the conduction of impulses through their axons in CNS. So they provide formation of myelin in CNS. It's their main function. And also they participate in the nutrition of neurons. It's another function of oligodendrocytes. So here we can see a neuron, and it's oligodendrocyte. It has long processes which surround axons of neurons. And here we can see astrocytes, which provide mechanical support for neurons. And third type of microglia, it's glia of inner covering of ventricles of brain and central canal of spinal cord. There are cavities inside brain and inside spinal cord, and they have special lining. Here you can see a cavity of ventricles of brain and its tissue of brain, and this is special lining, and there are ependymocytes and ependymoglia. There are ependymal cells which are a little bit similar to epithelial cells. They are tightly located to each other, but the main difference is absence of basement membrane and long processes which are beginning on the basal pole of those cells, and those processes are going deep in the tissue of brain. So cells which provide lining of the ventricles, ependymocytes, they are like epithelial cells, but they have long processes on their basal pole, and basement membrane is absent. They provide barrier between cerebrospinal fluid inside the ventricles of brain and tissue of brain, and also some oligodendrocytes, some ependymocytes, they provide synthesis of cerebrospinal fluid, which fills the spaces inside the brain. Here we can see central canal of brain, and there are ependymal cells which provide lining of the central canal of spinal cord, and also they provide lining of the ventricles of brain. Here we can see the shape of those cells. They are like epithelial, they may cuboid or columnar in shape, but their basal poles, they have long processes which are going deep in the tissue of brain. And here you can see ependyma of brain, it's tissue of brain. There are nuclei of glial cells in other glial cells. And here we can see ependymocytes which form this barrier. And also we have choroid plexuses where capillaries are present, and they are covered with ependymal cells. This choroid plexus, it provides synthesis of cerebrospinal fluid. Blood from capillaries is filtrated, and it goes to the cerebrospinal fluid, which fills the spaces inside the brain. And here we can see microscopic structure, microscopic slide, which shows us structure of typical ependymal cells. And at choroid plexus there are capillaries with blood, and they are covered with special ependymal cells. They provide filtration of blood plasma and synthesis of cerebrospinal fluid. So there are kinds of microglia, astrocytes, oligodendrocytes, which form myelin. Also, there are ependymal cells. And another group of glial cells, it's microglia. The main difference, principal difference, why do we classify macroglia and microglia into distinct groups, is that they have different sources of the development. Macroglia, it develops from neural tube. It has common source of development with neurons. And microglia, it develops from monocytes of blood. There are cells, monocytes of blood, kind of leukocytes, and they form different macrophages in different tissues and organs. They also form osteoclasts in bones, and some monocytes from blood, they migrate into CNS, into nervous system, and here they become to be microglial cells or microgliocytes. Here we can see them. So here we can see microglial cell. It has branched cell-like shape, and they are inactive. During activation, they become to be like a leukocyte. And the main function of microglial cells, it's defense, immune defense inside CNS, inside nervous system. So if some harmful substances enter in brain or some inflammation is taking place, microglial cells, microgliocytes, they are activated and they participate in the immune reactions. And here we can see activation of microglial cell, and it provides phagocytosis inside the brain. And also they provide phagocytosis of dead neurons. You know that neurons during our life, they are dying, and dead neurons, they don't disappear, and dead neurons may be harmful for neighboring neurons. So microglial cells, they are activated and they provide phagocytosis of dead neurons and other glial cells. So they provide cleaning inside the brain. Okay, so students, we have discussed glial cells, and now we are going to nerve fibers. And you should understand what are the nerve fibers. So here you can see different neurons. They have axon, each cell has axon and different number of dendrites. And perikaryones, they are located usually in CNS, but they also may be found in some structures of peripheral nervous system, in nerve ganglia of peripheral nervous system. And their processes, axons and dendrites, they excite from nerve ganglia, they excite from spinal cord, and they form nerve fibers. Here we can see nerve fibers. And what are the nerve fibers? Nerve fibers, they include dendrites or axons, which also have some envelopes, some covering. And here we can see axon with myelin covering, and we call this nerve fiber. So nerve fiber, it includes axons or dendrites of the neurons with some envelopes. And they aren't distinct structures, distinct fibers like collagen fibers. They are parts of neurons, dendrites and axons. But dendrites and axons, they have covering. They are covered with special cells, with special membranes, and it's necessary for their functions. So let's go to nerve fibers. And structurally, nerve fibers are of two types, myelinated and unmyelinated nerve fibers. There is special covering myelin. Myelinated nerve fibers, they have myelin. Unmyelinated, they don't have myelin. But also they have some covering, and it protects them. So let's begin from unmyelinated nerve fibers. They have a little bit simpler structure. And then we'll talk about myelinated nerve fibers. So here we can see cross section of axon or dendrite. It's called central cylinder. Here we can see axons or dendrites, usually axons, but sometimes there are dendrites. Here we can see it. And here we can see cell. It's special glial cell which is called Schwann cell or neurolemmocyte. its nucleus, its cytoplasm. And we can see here that this cell uptakes axon or dendrite. It's not phagocytosis. This cell, it surrounds axon or dendrite, and it forms cover and it protects this axon or dendrite. Here we can see, and here it completely surrounds axon or dendrite on this picture, and two membranes on two sides, they communicate. Here we can see incomplete, and here they completely communicate with each other, and this region, it's called mesaxon. And this nerve fiber or axon or dendrite is completely surrounded with Schwann cell, neurolimocyte. It provides protection and it also maintains this nerve fiber. And later, some processes, some dendrites and axons are surrounded with the same cell. Here we can see cell, Schwann cell, its nucleus, cytoplasm, and here we can see axons or dendrites. They are surrounded with one Schwann cell. So it's formation of unmyelinated nerve fibers. It has some axons and dendrites in one Schwann cell. So they form bundles, and they haven't complete isolation from the neighboring cells and neighboring axons and dendrites. And here we can see Schwann cell and axons or dendrites which are included in the cytoplasm of nerve cell, of Schwann cell. Here we can see this cell, and axons or dendrites are present inside this cell. And here you can see mesaxons, double membranes of Schwann cell where they communicate with each other. So it's structure of unmyelinated nerve fibers. And microscopic picture of unmyelinated nerve fibers. Here we can see axons or dendrites which are surrounded with Schwann cells, and there are nuclei of Schwann cells. They are present here. So bundles of axons and dendrites and nuclei of Schwann cells or neurolimocytes. And microscopic slide of unmyelinated nerve fibers. And let's go to myelinated nerve fibers. And beginning of their formation is the same, exactly the same as in myelinated nerve fibers. Schwann cell also surrounds, it uptakes and surrounds axon or dendrite. But here we can see principal difference. In unmyelinated nerve fibers, some axons and dendrites, they enter in the same Schwann cell, but in myelinated nerve fibers, only one nerve fiber, only one axon may enter in the Schwann cell. And here we can see that Schwann cell is being rotated and it surrounds axon or dendrite, and it forms some routes which surround this. And these circles of membrane of Schwann cell, they form myelin. Here we can see myelin layer. It's formed by Schwann cell. So in this case, some axons, they enter in the same cell. In this case, one cell is rotated above one processes. It's myelinated nerve fiber. And here we can see axon, and its Schwann cell, it's rotated above axon, and one Schwann cell can't cover entire axon of the nerve cell. So there are following Schwann cells which cover this axon, and gaps between them are called Ranvier nodes. Here you can see Ranvier nodes or nodes of Ranvier. They are places where neighboring Schwann cells touch each other. And here we can see Ranvier node, it's axon inside, and routes of the cell membrane, and it's microphotograph, electron microphotograph, it's cytoplasm of axon, and numerous layers of myelin, it's cell membrane of Schwann cell. and in the peripheral part of this layer there is nucleus and cytoplasm of Schwann cell. Here we can see it. Nucleus of Schwann cell, cytoplasm, and numerous layers of the mesoxone of the membrane of Schwann cell. So it provides very good isolation of nerve fibers, and also it participates in the conduction of nerve impulse. Here we can see Ranvier node, its axon, its myelin, its Schwann cell nucleus, and cytoplasm of Schwann cell. And also there are in the series of Schmidt-Lantermann, they are inclusions of cytoplasm between the layers of myelin. So it's principal difference between unmyelinated and myelinated nerve fibers. Beginning of formation is the same, but results are different. In the unmyelinated nerve fibers, usually there are bundles of axons and dendrites inside one cell. In myelinated nerve fibers, only one axon or dendrite may be surrounded with one Schwann cell. So here we can see that those axons or dendrites are isolated better than in this case, and each axon and dendrite has own covering, and it provides specific conduction of the nerve impulses. Which nerve systems or nerve organs have those fibers? Usually almost all fibers, they are myelinated, but autonomic nerve system, it has unmyelinated nerve fibers, and they provide slow conduction of nerve impulses. Here we can see unmyelinated nerve fibers and myelinated, and there are Ranvier nodes between neighboring Schwann cells and routes of myelin which cover axon. And here we cannot find myelin because this Schwann cell cannot be rotated above all those axons. Unmyelinated nerve fibers, they provide slow conduction of impulses. Impulse runs continuously without any gaps, and it's slow conduction. Wave of depolarization runs on the plasma membrane of axon or dendrite in the some direction. In the myelinated nerve fibers, it's saltatory type of the conduction of nerve impulse. Nerve impulse jumps from one Ranvier nodes to another, and it provides very fast conduction of nerve impulse. Here, in unmyelinated nerve fibers, conduction of nerve impulses is very slow. In myelinated nerve fibers, conduction of nerve impulses is very fast. So its main difference functional between two kinds of fibers. And here we can see microphotograph. It's a drawing of histological slide of myelinated nerve fibers. Here we can see axon or dendrite, its central cylinder, it's myelin, it's Ranvier node. There are incisors of Schmidt-Lantermann, and it's cytoplasm of cell of Schwann and nucleus of Schwann cell. Its structure of myelinated nerve fibers and their appearance under microscope. It's microphotograph of myelinated nerve fibers. Here you can see bundle of myelinated nerve fibers and their outer covering, it's myelin. And cross section of the nerve, and here we can see myelinated nerve fibers, myelin, and central cylinders are light because special staining is applied which reveals lipids in myelin. Myelin is rich of phospholipids, and special staining reveals different fats and lipids in their myelin coverings. And in peripheral nervous system, Schwann cells, they provide formation of myelin. But in central nervous system, there are special glial cells which are called oligodendrocytes, oligodendroglial cells, and they provide formation of myelin in CNS. So it's difference between PNS and CNS. So what is another difference? In peripheral nervous system, Schwann cells are completely rotated, and they completely surround nerve axons or dendrites, And in the CNS, you can see that cell body of oligodendrocyte isn't surrounding axons or dendrites. Only processes of oligodendrocyte, they participate in the formation of myelin. That's why one oligodendrocyte can participate in the formation of myelin of some neighboring neurons, some fibers. Here we can see this segment of the fibers, this and this, they are formed by the same cell. But in this peripheral nervous system, one cell may participate only in formation of one nerve fiber. Its main difference between peripheral and central nervous system. So in central nervous system, oligodendrocytes participate in the formation of myelin of nerve fibers. In peripheral, Schwann cells, specialized Schwann cells, which also are glial cells, but glial cells of peripheral nervous system. Here we can see that nerve cells, they generate impulses and they send them to another cells or to effector cells via nerve fibers, and they somehow conduct those impulses to another cells. And there are special nerve endings and interneuron communications which provide this communication between the neighboring cells. So what are the nerve endings? Nerve endings may be afferent, may be efferent, and also or sensory and motor, and also they may be located between neurons, so they may be associative, and as well as nerve fibers. Nerve fibers also may be in their by their function, they may be sensory, they may be motor, and also they may be associative nerve fibers. So the same classification may be applied in to classify three structures: neurons, nerve fibers, and nerve endings. They may be sensory, associative, and motor. And now let's go to the nerve endings and interneuron communications. We'll begin from associative nerve endings, and they are known as synapses. They provide communication between neighboring neurons. And synapses, they may be, according to their localization, they may be axosomatic between axon and body of neurons, axodendritic between axon and dendrite of neuron, and axoaxonic between axon and axon of another neuron. It's classification according to their localization. Also, synapses may be chemical and electric synapses. In chemical synapses, special neurotransmitters, chemical substances, they provide conduction of this information. In electric synapses, electric wave directly runs from one cell to another. They are very located very close to each other, to membranes, and that's why neuromediators aren't needed. So let's look at structure of typical synapse, and here you can see axon ending. It's a membrane of axon. It's presynaptic membrane, and there are synaptic vesicles with neurotransmitter, and so synaptic vesicles, they bring neurotransmitter, they uptake neurotransmitter which was synthesized in the cell body, and special axon transport provides it carries neurotransmitters to the axon ending. And here we can see synaptic cleft. It's presynaptic membrane and postsynaptic membrane. It's those membrane which accepts information from previous cell. And it's postsynaptic membrane, and it's some part of the cell which receives information. For example, it may be cell body of the neuron. It may be axon or dendrite of another cell. So presynaptic membrane, postsynaptic membrane, and synaptic cleft between them, and there are vesicles with neurotransmitter in their axon ending. And now let's look how does it work. Here we can see nerve impulse. It runs on the cell membrane, and it's excited cell membrane of the axon, and it causes exocytosis of the vesicles with neurotransmitter, the synaptic cleft. There are special chemical substances which are released in the synaptic cleft. And those substances, they are attached to the receptors on the postsynaptic membrane. Here you can see receptors, and they are activated after this. And after this, it causes generation of new wave of nerve impulse on the postsynaptic membrane. Here we can see generation of new wave of nerve impulse, and this cell has generated own new nerve impulse after the action of the synapses. So how does the synapse work? And there are neuromuscular synapses between nerves and muscles. They send impulses to the muscles, and here we can see axon endings on the nerve fiber of the muscle fibers. And you know that each muscle fiber, it has own source of innervation. And here we can see how nerve impulse comes here, and it's synaptic cleft between axon and muscle. And also there are sensory nerve endings. In muscles there are motor nerve endings, and also there are sensory nerve endings. They are present in different organs, and also they usually are present in skin. They react to different information from outer environment. There are free nerve endings which are freely branched in the epidermis of skin. Also there are encapsulated, non-encapsulated nerve endings, and we'll study them later in the following topics. Here we can see skin, and there are different nerve structures, sensory structures, which react to touch, to vibration, to temperature of outer environment. So there are sensory nerve endings, and dendrites. They receive information from sensory nerve endings, and they carry impulses to CNS.